Please welcome Bobby Sandage, Lightchain Capital.
Ladies and gentlemen, it's my profound honor to introduce Dr. Chris Bahl, a gifted scientist and protein engineer whose innovative work has consistently pushed the boundaries of what we thought possible. Chris's work focuses on a special type of protein engineering called de novo design, which is where computational modeling is used to design completely new proteins from scratch that have amazing properties that evolution could never create. Chris came to Boston in 2017 to join the Institute for Protein Innovation at Harvard Medical School as one of the founding faculty members. He built a thriving research lab and attracted top talent to join him. Then in 2021, Chris founded AI Proteins, quit his job in academia, and canceled all of his research grants. His whole lab then joined him at AI Proteins. Chris and his team are at the forefront of amazing discoveries of new, innovative medicines. His leadership at the company continues to inspire and remind us of the endless possibilities that lie ahead. Please join me in welcoming the extraordinary Dr. Chris Bahl.
Breaking the wall to AI-driven protein medicines. Chris Bahl, AI Proteins.
Communication is vital, and this is true at all scales. What we can see here is a painting of two cells having a conversation. They're cells in the immune system, and they're discussing that little red bit in the middle, which is a piece of a virus, and they're trying to decide through this conversation what to do next. Now, the language of cells may be unfamiliar to us, but it is a language nonetheless. Each of these colored blobs is a different protein, and these proteins have handshakes, and the strength of these handshakes, the duration of them, all transmits information. Now, most of our drugs that we use today to treat diseases work by interacting with one protein, and you can see many proteins involved in this discussion. It's the linguistic equivalent of only knowing one word. You can communicate a little bit and sometimes get done what you need to get done. But it's still fairly primitive compared to what's possible if you're fluent in that language. And so this is what we're trying to do, become more fluent in the language of cells to create a next generation of medicines that are safer and more efficacious. So when you want to communicate, the first thing you need to be able to do is have this foundational vocabulary. And so we have a lot of different words that are able to provide some instruction. And using generative artificial intelligence, we create proteins that effectively communicate these words. But as you know, the dictionary is not a riveting read. You need also grammar to put them together, to create phrases, to provide more detailed instructions. And here now we're providing an instruction for an immune cell to go and attack a specific cancer cell, and giving it more context on how it should do this. This type of approach for creating new medicines is not possible using natural proteins. So we need to build them from scratch in order to be able to confer these molecules with the properties that they need to tolerate all these types of fusions. Natural proteins will fall apart. So here's really the breakthrough, is the de novo design of a new modality called a mini protein. It's a peptide that folds into a 3D shape, and that 3D shape makes them truly remarkable. They get to bind to their targets when we engineer them really precisely, and that makes them very safe and specific. We can link them together to create these phrases, these instructions, change the order in which they appear in the chain, the number of times that they are there. And importantly, we can do all of this using amino acids, the same natural building blocks that all of our proteins are made of. So this makes our medicines safe and fairly straightforward and cost-effective to produce using bioprocess methods. We are called AI Proteins, but we do a lot more than just AI. We combine synthetic biology and robotics, and we've created an incredibly high-throughput platform for generating novel protein therapeutics. The company's just about 3 years old now, and we've drugged about 140 targets to date. This is way more than we could ever take into the clinic ourselves. our main program that we are pushing forward into the clinic is a T-cell engager for lung cancer. This is a massive market, $20 billion a year drugs for T-cells, and 2 million patients globally diagnosed every year. We also partner with pharmaceutical companies as well to bring additional drugs into the clinic. Really, our mission is to make as many new therapies as possible and to get them to patients as quickly as we can. And so this means working with others, and this is a large part of our business model. I'm also incredibly lucky that my lab came with me to this company. We've also grown. We combine a lot of expertise in software development, in drug development, protein biochemistry, biophysics, and layer on top of that business development, accounting, all of the bits and bobs that you need to make an effective company. And thank you all. Happy to take any questions.
That was fast. You are the fastest so far. Actually, we had 30 seconds. I would love to talk with you about synthetic biology linked to robotics, but we have the jury asking questions first.
Please.
Okay, so let's go with the questions. We will need to have the timer back on 4 minutes, please. And then we can take it from there, or shall we? Yeah, okay. We have, sorry, the juries first. We have here, yes, exactly.
Great presentation. How do you think about the long-term strategy? Like, will you continue to stay a platform company and partner with many organizations, or will you also kind of become a full-fledged therapeutics company at some point and try to capture more value?
Yeah, so probably, I mean, 2 exits, obviously, IPO or acquisition. There may be a path where we become a clinical, like a commercial company where we're selling drugs. I think far more likely is that we'll push our drugs through value inflection point, phase 1 or phase 2 clinical trial, and then license it to a large pharma and keep doing it. My passion is really the platform and creating these medicines. And I think so often you see the platform being cannibalized in order to fund the clinical asset. This is truly a fundamentally new way of making medicine. And I wouldn't want this to get to end too early before we get the full potential of this out into the world to do the most good.
Okay. Next question was Frank, and then I don't forget you. I see you, but I give prior to the jury. Frank, please.
Yeah, thank you very much for the presentation. I've not really fully understood how you created predefined libraries, the first question. And the second question is this based on the natural ligand or based on the natural binder, whatever? And the second question is how you build in CMC characteristic and pharmacokinetic characteristics.
So these proteins are generated using AI. So they don't have any natural basis. They're completely new. So when we functionalize them to bind to a target, we do this in a couple of ways. We can design them specifically to interact with a target site that we know where the drug needs to interact. And we have a second approach that's sort of like a fancy new way of doing an old-fashioned idea, which is to generate large libraries. But our approach, there's no randomness in the libraries. Every member is designed to be a drug. We just didn't design it to a specific target. And then we can use the synthetic biology approaches to fish out the things that work. And this actually allows us to uncover novel disease biology, which has been really a powerful tool. The second is, in your question, is around manufacturing and biodistribution. So one of the very cool things when we design these structures is they really are whatever we want them to be. So one of the things that we want them to be is extremely stable. So they spontaneously fold themselves up, and when they do so, they become extremely durable. So we can boil them, you can dry them down, you can freeze thaw them, all sorts of like organic solvents, all sorts of horrible stuff you would never do to a typical biologic drug. These things can withstand no problem. And when we want to modulate the PK, biodistribution, we have the ability to continue to link these things together seemingly ad infinitum. And so we've made additional modules that we can attach into these, which will extend the serum half-life. If we want the serum half-life to be short, we can leave those out. Depending on the indication and how you're trying to use the drug, you may want different approaches. If you're sticking like a radioactive nuclide on it, you want that to clear the body fast. If this is, say, an anti-inflammatory medication, you want it to hang around in the blood for extended periods of time.
I'm sorry, at the back, we won't take your question. We have 30 seconds, so very short question, please, very short answer.
Thank you very much. You mentioned that you're using AI to generate these. For using generative AI, you typically would have to train that AI on something? What is the base data that you're using to generate these?
15 seconds.
The protein data bank and a massive pile of proprietary data that we've harvested ourselves.
Okay, thank you very much. That was it. That goes fast. Woo.